Cytomine is a collaborative biomedical imaging and mining solutions that focus on histology and pathology, which are the science that focus on observations of cells and tissues with a microscope. The work of a pathologist or a histologist can be a lonely one. The samples is placed on a thin slide of glass that is very fragile, so when the pathologist needs a second advice from a colleague, he will definitely avoid to make this slide travel. The risk is too high to have it broken. When the first robotized microscope and the first wall slide scanners appeared on the market a decade ago, the dream was that transforming these slides in images will make collaboration between pathologists easier. But digital pathology images are not as simple to share than classical imaging ones, and here is why. The main difference between the two, beyond the fact that the medical imaging is in vivo and the pathology is in vitro, is the resolution at which the images are taken. In medical imaging, the resolution is quite low, of the order of 1 mm per pixel. In digital pathology, we work on scales of the order of the micrometer or the nanometer per pixels, as the aim is to mimic the microscope. The consequences is that the digital pathology images are of enormous size, both in surface and volume. Images of more than hundreds of thousands of pixels per size and nearly 100 gigabytes in volume are not uncommon, just for one image. When in medical imaging, we rarely exceed 1,000 pixels and 10 mega in volume. If we compare two classical images in both technologies, as a human chest X-ray on one side and a slide of lungs of mice on the other side, we will see that we will need to use 625 X-rays to have the same surface of one histological sample. In terms of weight on the disk, as digital pathology is in 3 colors channels, it means that the one digital pathology image will occupy on the disk the same volume than close to 2,000 X-rays. Due to these limitations, it's quite easy to understand why digitalizing the slides was not sufficient to enhance collaboration between pathologists, as sharing this image over internet was really a challenge. But if Google succeeds to share images of the Earth using Google Maps, why not trying to use the same kind of technology to share digital pathology images? That was the idea that drives the development of Cytomine since 2010, to move all the technology part from computers of the pathologist to web servers and to develop a full web application to share but also to annotate whole slide image over the web. Using Cytomine, pathologists are able to work in full remote at any time from anywhere. The platform has proved to be able to allow collaboration for small teams like tumor boards, but also very big groups, as the record is actually 4,000 people on the same images during a MOOC. We will not spend too much time here on the technical assets of our solutions, but we will limit ourselves to mention that Cytomine has been developed by pathologists for pathologists, and that we have been really focused on building an application which is collaboration friendly. Cytomine has also been developed since the beginning as a platform to help research in artificial intelligence applied to digital pathology. So we consider a platform to be also AI friendly, as it allows you to use but also to develop your own AI and to train it within the same tool. Here on the right you can see the dashboard of Cytomine. Using our web viewer, you will be able to explore your whole slide images as if you were using a microscope, but also to annotate them and to add some semantic information to these annotations. These annotations can also be created by your AI, as here, where an AI has segmented the zone where there is a high probability to be a carcinoma. One of the assets is a Cytomine Basic Edition is open source. You can install it and use it for free, including the AI engine. When it comes to more specific needs, the Cytomine company sells some services and products to help enterprise to reach more ambitious goals. Here are some examples of such activities. The first one is teaching. The aim here is to allow huge groups of students to have access to not so big collection of images to help them to learn to recognize healthy and unhealthy cells and tissues. One example of enterprise module here is a connector to the learning managing system of the university. The second example is research. We consider here research done at university, but also in companies like CROs and pharma biotech. In such institutions, which are doing research in pathology but also research in AI applied to pathology, the challenge is to mine very big collection of images based on the collaboration that can imply engineers if it uses artificial intelligence. The challenge here is the amount of images. An example of enterprise modules will be a connector to fetch the image from the laboratory information system and to push back into it the results of the different analyses. The next step for Cytomine Enterprise Edition is the routine diagnostics at hospital, to allow pathologists to collaborate with colleagues worldwide, but also to benefit from AI to help them to do more quick and accurate diagnostics. One of the challenges here is to manage patient data which must be stored in the hospital information system. But the real future of Cytomine is to provide validated AI dedicated to all these sectors in a centralized Cytomine AI store. This AI store will deliver AI developed by our teams, but also by validated partners. If you want more information about Cytomine, please consider to visit our website at cytomine.com.